Thanks, Emily. So yeah, I'm a pacer, so this is actually difficult for me to be nailed down to a spot, so I might end up wandering from here. So I actually thought I might get the Traveled the Farthest award being from Maine, but Paul ruined that for me. Thank you very much, Paul. And I also did want to say that, you know, I love coming to these conferences every 3 years. You really feel like my people, and a great example of that came up today when Emily was talking about something related to fairy rings and about how some of them were not actually part of the same genotype, and there was a mumble that went across the room. And I thought, yes, okay, everybody thinks about these things like I do. They're fascinated just as I am. Well, so I'm going to talk to you today about clonality and epigenetics, and I want to start by saying if you're expecting data, there's way out right now. I'll wait a couple of seconds for you to make your exit, but we're really just starting this thing up. So I'm going to spend some time really talking to you about what we're thinking, where we're going, and possibly what this might tell us about this really growing new field in ecology and its relevance to redwoods. Starting there, I'm working with Richard Dodd at UC Berkeley, and I just have this little figure of a clonal plant to remind people that a clonal plant or a clonal tree is the same genotype represented multiple times across the landscape. There are many ways that can happen, but I don't need to explain to you guys that that's fairly common in redwoods. All right, so just briefly, some of the work we've done in the past on clonality, not epigenetics. This work was looking at the structure of clonal growth within second growth stands. So Deb Rogers looked at it within old growth stands. We looked at it within second growth stands. And sure enough, we found that clonal growth was important. There was quite a bit of it going on. And that the vast majority of stands were represented by a very small number of clonal individuals. So of actual genotypes. So you can see down on the bottom right, the wedges that are pulled away are the single stem genotypes that we found, and the other wedges represent the remaining genotypes that represented the rest of the individuals on the stand. And you can see that there are many situations where there may be only 4 genotypes, and they're representing up to 80% of the stand itself. And in addition to clonality, in terms of the numbers and size, we also found that it was quite variable, that it was variable in structure, and so it was difficult to predict what clones looked like. And it was difficult to identify unless you actually got in there and did the genetics. So we found, for example, not just fairy rings, but we found fairy ring chains. And so that's what you see on the right-hand side there. It's one genotype. The gray, the filled-in dots are a single genotype, and it is represented in multiple fairy rings. Then we have these concentric circles where there are fairy rings, and then there are larger fairy rings outside those fairy rings. And then there are sometimes where we just can't even find a fairy ring, where they're just all over the place, and it just looks like a stand without fairy rings. Our suspicion actually is that, remember, these are second growth, so our suspicion was that maybe that was slash in effect that had come up, and that these were just spread out all over the place. And then finally, there was the instance of great distances, 40 meters, and actually it could have been more than 40 meters. That's where our sampling ended. So we don't actually know what the maximum could be in a clone. When I talk about clonal plants, clonal trees, I don't want to give the impression that they are only clonal, right? We've heard already today that there are seeds being produced out there, and actually it's great to hear that they are more viable than what I had thought historically. And so there is sex going on too. And it's actually suspected that there's no strictly clonal, there's just something on a spectrum. And redwoods lie somewhere on that spectrum. They're not only sexual, but they are somewhat clonal under certain circumstances. And so we spent some time trying to figure out, why go clonal? If you can do both, why go clonal? And there are some great advantages to being clonal. You have the possibilities of things like resource sharing, especially if there's integration that remains, risk sharing. If you have multiple stems, some calamity occurs, the chances that at least one of the representatives of that genotype will survive is fairly high, and so you don't lose that genotype from the area. Division of labor, everybody can share, for instance, one large root system, and so the economics make a lot of sense. Reduce reproductive risk, right? You're getting this nursing effect from stems that already are in the canopy, and then longevity. Really, how long do these trees live when they can keep on, regenerating the way that they do? So those seem like some great advantages. There are disadvantages, though, right? I mean, if you're not doing reproduction by seed, then you're really limiting your ability to spread. But even more important, and this really is the big one that leads people to wonder, why are we even seeing clonal growth out there? It doesn't make sense according to Darwin, right? We have very little capacity, or there's a very slow rate of reproduction for adaptation. You're not getting a lot of innovation going on out there. So how is it that this species can live so long, cover such great area as clones, and experience this heterogeneity both in space and time? And how does it do that? How does it manage that problem without having adaptation? So that's what gets us to epigenetics. Epigenetics is this new, relatively new to ecology. It's been talked about quite a bit in other fields, and ecologists tend to be late to the game. But you'll hear a lot of talk about this, but there haven't been a lot of studies in woody plants yet. We're really sort of on the front edge of that. Now, so what is epigenetics? Epigenetics are changes in the way that the genes are regulated in response to the environment. But, in addition to that, that's actually not represented by sequence changes in the DNA. So we're not talking about genetic variation. We're not talking about diversity, We're talking about how the genes are used differently. And the interesting thing is that there's a recording mechanism in effect that allows that response to be retained, the information in effect to be handed down to the next generation. And that's true both in mitotic or meiotic generations. So that can be through clonal or through seed reproduction. Now, there's differences in how much that retention occurs when you're going between mitotic and meiotic. So I don't want to give the impression that, all information is transferred, but the fact that it even occurs is really pretty amazing. Because you're not having to go through adaptation, you can adjust, right, fairly quickly. So we're starting to talk about plasticity. Your ability to acclimate can happen quickly. It also can occur, it can build over time, which allows for more fine-tuned responses. It's not just on-off. And so, for example, if conditions persist, then responses can build, right? And it's clear that that can be a huge advantage. And it's reversible. So that means that certain responses can be tuned to the types of stimulus and how often those stimuli come and go, and the ability then to be nimble on the appropriate time scale. All this makes a lot of sense when you're talking about a redwood, right? Talking about living incredible long expanses of time and space. And this fine tuning is really going to be important when you are experiencing fine gradation across the landscape when you're represented multiple times as a clone, right? This is the thing is, right, remember, within the context of a clonal species, there's the identical genotype each time, but that genotype is not experiencing the same conditions each time, and so in order to optimize, it needs to be flexible. Okay. So when I think about the range of, well, where epigenetics fit within the range of adaptation to acclimation, you have your normal, natural selection and so on the left-hand side there, and then you have your physiological responses on the right-hand side, and epigenetics somewhere in between. It's not one or the other. It has some of the character of both of those, right? It has a quick response time. You don't need a new individual. You don't need the original individual to die. But it has some of the characteristics of adaptation because it actually can be recorded and it actually can be handed down. So there's this interesting in between that can be quite useful, especially if you're clonal and you're not doing that far left quite so much. Now, Richard and I have been spending some time thinking about what it means to be clonal and epigenetic, and I've already mentioned several times that the spatial heterogeneity, the ability to adjust to temporal heterogeneity and progressive, and then also many of you are aware of this whole idea of priming, and if a plant experiences something, how often the second time around or the third time around the response can be quicker, right, or more effective. A lot of you have seen this happen, and so really, how far off is that from learning, in effect, epigenetics as a mechanism of learning? And so that's what gets us to this, which is, this is what clonal ecologists are doing right now at the clonal conferences. Just take a minute and think about this sentence, right? A colleague of mine from the Czech Republic wrote, which is the more faithful inheritance of epigenetic information in clonal plants, in conjunction with information exchange and coordination between connected ramets, is likely to enable especially advanced intelligent behavior, right? I mean, we're talking about plants, right? That's not even supposed to compute. And of course, they're being a little bit loose with the vocabulary, but you get what they're saying here, right? Is that there are mechanisms to do some of the things that look like memory, that look like learning, that possibly even look like intelligence. That when you have a network of individual stems that are sharing information with each other, and each one of those stems is storing information, well, it doesn't seem like the jump is that far. And that's pretty fascinating when you're talking about an organism that lives as long as Redwood does and can learn a lot in a long time. So what are we doing? Well, because we're at the beginning here, this is step one, what we're doing is we're interested in understanding where this variation is. And we know that there's going to be variation within an individual ramete, within an individual stem. And so really one of the first steps we're looking at right now is from top to bottom, trying to map out where that variation is, what these epigenotypes look like, and where's the variability. But then we also want to understand within a clone, and then between genotypes, and then finally sites, right, and across the distribution. And so hopefully an understanding of that better will give us a chance to predict a little bit more, and I'll talk in a bit about where hopefully that'll take us. What we're doing is we're using a relatively new technology. We've got collaborators in the Netherlands who have developed this technology, EpiGBS, with the epi obviously is epigenetics. and the GBS is genotyping by sequencing. And what's great is that making the jump to sequencing allows us to get really specific about what we're looking at. Up till now, microsatellites have been very helpful, for example. We've done a lot of work with them, but the chance to be able to look at the actual sequences and start to figure out where those lie within the genome, and as Emily described earlier, that sequence will be available hopefully soon. That's going to be fantastic. We'll be able to maybe zero in on the specific genes that are responding to different conditions, and this might be a way for us to zero in pretty quickly on which are the ones that are, for example, turning on and turning off a lot, right, and playing a big role. And then finally, this bisulfite sequencing approach is really useful because what it does is if you have your DNA, the mechanism that we look at, there are many, but the mechanism we look at for epigenetic control is when a methylation occurs, it's basically a brick that sits on top of the DNA and doesn't allow that portion of the DNA to be read. And so what we're looking for in the epigenotype is where we're seeing methylation events or not, where things are being turned off or not. And the bisulfite sequencing is really helpful because what you do is you change all of the C's in your DNA to U's, and the change is not allowed to occur if there's a methylation on that location. And so what you can do is you can compare individuals and see, oh, well, there is variation here. And actually, let me just jump ahead and show that. In this lower right-hand figure, you can't quite see the colors too well, but you can see there's a red and blue, and then you can see there are places where the T's appear and disappear. And so what that's telling us is that there's variation at that site in terms of the methylation, and we can start to quantify that. And this is one thing that's really cool about this also is that, plasticity has been a difficult thing to quantify. What we've really been doing is we've been quantifying plasticity in a particular trait, right? So we identify this trait and say, okay, fine, it's changing this much or not. But what we're not really looking at are all of those components internally that are propelling that response, right? And so here we are actually at the root of it all, being able to put a number on how much variation is actually occurring. For example, if you don't see any change, right, that often can just be homeostasis, right? But internally, there could be a lot going on to maintain that homeostasis. So here we are, we can get inside and see what those responses are. On the upper left-hand side here is some of the early data, and this is all you're going to get to see, so don't expect any more. 2 individuals, and what we're seeing here is that we are able to pull them apart. We're able to pull the epigenotypes apart. We actually do see variation, so it looks like it works. And now we want to do this on a large scale. We want to look at hundreds of samples, start by looking within genotypes, and work our way outward. So, there are many directions this could go, and there's plenty of room for everybody to jump in. So please, let's collaborate. Let's get stuff happening here because it's really fascinating stuff, and I think it can be really powerful. For example, better understanding the reversibility of some of these responses could be incredibly helpful in understanding how stands might respond to climate change, right? Especially if we're not looking at sexual reproduction. We're looking at the same individuals who may be here and would have been here another 1,000, 2,000 years. And if we have some sense of what that reversibility might be, then we have a sense of how much they might be able to cope with in terms of future conditions. But all these things are open. We don't know the answer to these questions. And what will be really interesting also is if we can start to get really specific about specific genes, and then the specific circumstances and how much of a response there are to the different stimuli. So finally, obviously some thanks. I want to thank CAL FIRE for funding a lot of the early research in the clonal work. Save the Redwood League has been great in helping us propel this epigenetics question forward. And of course, I couldn't do any of this without my clan of crazy students who make it interesting. So thanks a lot. Yeah, Chris.
If it's not proprietary information, tell us what kind of site looked at.
Well, so at this point right now, we're trying to figure out what those, so we are at the stage right now where we're looking at different enzymes and trying to figure out which enzymes are going to give us the most variable loci with the least coverage per. So we want to spread out our sample size as much as we can and get as much information as possible with that, right? And we're still trying to balance that equation. Yes, so what happens is that you've got your entire genome that then gets reduced by the enzymes that cut it down to some manageable size, and then within that manageable size, then you do your bisulfite conversions, and you look at the sequences there, and that helps you build some loci, some particular loci that you're interested in, and then eventually you can tie that to the genome. Not well. We don't know what they are in terms of where they are, right? Because we don't have a genome yet, right? And so yes and no. We know what this loci is. We identify it, but we don't actually know because we haven't built it yet. We haven't turned it into. But we're going to hopefully be able to do that, right? Yeah.
So this kind of goes back to something that you pointed out at the beginning about the advantages and disadvantages of cloning. And my coworkers and I have been posed this question, we've been asked this question a lot, and we get kind of stuck trying to answer it. But when we think about in regards to climate change, in regards to stressors, drought, when you try to clone the results from your phase, like you're talking about the phase stress.
Right.
Let's say if you're talking specifically about drought, and they have a whole bunch of issues, a whole bunch of new clones coming out. How is that an advantage if now they are competing for resources? Are they competing for resources? For water, there's a time drought.
Right. So I think if you're talking about just sort of like your typical small sprouts, right? I don't think that probably not that big a deal, right? There's in terms of the biomass, is pretty small relative to the stem, right? But at some point they do start to have an impact. Now, when that is, I don't know, maybe at that point, maybe you've got somewhere between 5 and 10 ramets or something like that. That's when you start to maybe see some kind of effect. I suspect, and people in the field can tell me, right? I mean, don't you see this more in situations where maybe there's an opening or maybe there's an opportunity and the tree has some added resources that it can take advantage of? And so maybe that's what it's doing, is it's sort of hedging its bets, right? It's like, oh, here's a chance. And this is part of that clonal spread thing, right? Clones are looking for opportunities. They're opportunists, and they want to move. And that's one of the really interesting thing about clones, right, is that we like to think of plants as being stuck in one place. But actually, they can be mobile. Like, they can move. They can abandon that rammet because they've set up 5 that are down here, and now they're in a better location, right? So I think a lot of it's about time. The time scale that you're thinking about, right, is not snapshot, and then, size, I think starts to play into that. Does that make sense? Does that help? Okay. Rainbow.
I just wanted to ask a question. So within this research framework that you're setting up, do you think there is space for understanding, like in special long-lived tree species like this Redwood, if somatic mutations are also potentially contributing to the evolution / adaptability of different ramets? Because if you're having basal sprouts that come out at some time, and then the apex of that being treated in 2,000 years different.
Right. Right. So I think the answer to that is that will come up because we're actually looking at the sequences, and so we'll be able to see if those occurred or not. Right. Actually, we can differentiate those that are methylation events and those that are mutations because we look at both the forward and the reverse, and if the C changes to a U, right, but then the G doesn't change, right, then we're probably not looking at a mutation. We're probably looking at a methylation, right, conversion. Whereas if that C, you see what I mean? If you compare the two ends, right? Yes. Yeah. Yeah.
So how,
Sorry.
no, very much.
Thanks.
So how common are we that methylation is the main form of inheritance of epigenetics?
Oh, that methylation is the mechanism? Is that what you said? Oh, actually, I don't think we have a good sense of it. What we know is that it's the easiest thing to look at right now. And so that's what a lot of people are doing, right? There are some cool tools to be able to do that, but there's apparently a whole bunch of mechanisms, and a lot of those mechanisms appear to be remembered or appear to allow for memory. So we're right at the beginning, I think, of this. Yeah.
So then why focus on epigenetic changes rather than somatic mutation? Because I would assume epigenetics is probably more expensive to investigate than just comparing the genomes.
Well, so for example, somatic mutations aren't going to be reversible as a response to a condition. So a stem, it gets one shot at that, but then it doesn't really get a chance to go back. There's a lot more nimbleness that comes from really maintaining the same tool, which is the gene, and then just using that tool differently as opposed to just trying to change the tool itself.
Yeah.